Hello, students. Welcome to week 6 highlights of advanced pathophysiology. This week, one of your readings was about immunity. The most important physiologic function of the immune system is to prevent or eradicate infections. Also, it prevents the growth of many tumors and cancers that can be treated by stimulating immune responses against tumor cells. The immune system also plays a major role in the repair of damaged tissues. Vaccination is the process of stimulating protective adaptive immune responses against microbes by exposure to non pathogenic forms of microbes components. For example, vaccination against smallpox was eradicated worldwide. In contrast to these beneficial roles, abnormal immune responses cause many inflammatory diseases with serious morbidity and mortality. The acute or inflammatory responses is a carefully mediated response to damaged tissues. It involves four cardinal symptoms: redness, swelling, heat, and pain. Acute inflammation develops as a consequence of innate activation. Mast cells, macrophages, and related sentinel cells detect tissue damage and invasion, rapidly initiating the inflammatory cascade. Host defenses are grouped under innate immunity, which provides immediate protection against microbial invasion, and adaptive immunity, which develops more slowly and provides more specialized defense against infections. Innate immunity, also called natural immunity or native immunity, is something you are born with. Despite the wide variety of microorganisms we are exposed to during our lifetime, the innate response is more or less the same against each microbe. It does not change or adapt in any way. For these reasons, the innate immune response is also called non adaptive or non specific immune response. These are the first line of defense against invading microorganisms. While the innate immune response will eliminate many potentially harmful infections, some infections take longer to control and eradicate. In these cases, the second line of defense is needed. The second line of defense is also called the adaptive immune response. The adaptive immune response will come into action within 4 days and provide a second defense line. Lymphocytes carry out the adaptive immune response with highly diverse and variable receptors for foreign substances, and the products of these cells are antibodies. The adaptive immune response is more specific and develops over a lifetime with each new infection. It is known as the acquired or specific immune response. The cells of the immune system are located in different tissues and serve different roles in host defense. Most of these cells are derived from bone marrow precursors that circulate in the blood called leukocytes. The main types of immune cells are macrophages. These are a mature form of monocytes that have migrated into the tissues. Macrophages are present in almost all tissues of the body. Macrophages play a role in both the innate and adaptive immune responses. They are phagocytic and secrete cytokines that trigger inflammation and activate other immune cells. Macrophages also clear dead cells and cell debris in their role in healing damaged tissues. However, macrophages produce much of the tissue damage occurring in chronic inflammation. Monocytes are a minor component of the white blood cell count. Monocytes quickly enter the tissues of the body where they develop into macrophages. Neutrophils are a significant component of the white blood cell count. The role is innate immunity, where they play a crucial role in acute inflammation. Neutrophils are recruited from the blood to the site of infection and are phagocytic. Basophils and eosinophils defend against parasitic worms and ticks, which are too large to be engulfed by neutrophils or macrophages. They are involved in allergy. They form only a minor component of the white blood cell count. Mast cells are found in many body tissues, especially just underneath the skin and mucosous epithelial surfaces. Like basophil and eosinophil, mast cells have a role in immunity against parasites. They, con- they contain granules of histamine. Mast cells are involved in acute inflammation and allergy. Lymphocytes are responsible for the adaptive immune response. These are There are two main types of lymphocytes: B cells and T cells. There is also a lymphocyte called natural killer cells or NK cells. These respond to virally infected cells and tumor cells. Lymphocytes originate in the bone marrow. B lymphocytes mature in the bone marrow and, on reaching maturity, are released into the blood. B lymphocytes are the only cells capable of producing antibodies. Therefore, they are the cells that mediate humoral immunity. T lymphocytes mature in the thymus gland during childhood and are then released into the blood. T lymphocytes are responsible for cell-mediated immunity. These cells are mature but naive. This means that they have just to encounter antigens. Antigens are molecules, usually fragments of invading microorganisms, which B and T cells recognize and respond to a part of the adaptive immune response. All mature lymphocytes circulate in both the blood and the lymphatic system. Lymphocytes are present in huge numbers in lymph nodes and lymphatic tissues where they are exposed to foreign molecules and are activated. Dendritic cells are present in these tissues and lymph nodes. Dendritic cells display or present fragments of the microorganisms on their surface. This display helps to activate T cells. As noted above, pieces of a microorganism are antigens, and for this reason, dendritic cells are called antigen-presenting cells. Langerhans cells are specialized antigen-presenting cells found in the skin. In addition to the immune system cells, it is crucial to be aware of an essential family of signaling molecules associated with the immune response, the cytokines. Effector CD4 T cells or helper T cells produce proteins called cytokines that activate B cells, macrophages, and other cell types. Effector CD8 T cells have the machinery to kill infected host cells. Examples of cytokines include the interleukins, interferons, and tumor necrosis factors. They generally act locally to affect the cells near them and work on more distant parts of the body. There have been many pandemic diseases throughout history, the most notable ones caused by viruses, including the influenza pandemic or the Spanish flu in 1918, and another in 2009 by the influenza virus H1N1. The recent outbreak of coronavirus disease in 2019 has resulted in a global pandemic. The immune system plays a vital role in the host defense against the infectious agents against these pandemics. The host can mount immune responses upon infection by viruses and other microbes and control these pathogens spread within the body. However, some viral strains can evade the immune attack and proliferate in the body and elicit inflammatory responses, signaling protein molecules that elicit massive inflammation, a cytokine storm, which triggers the lungs, attacking different tissues, resulting in pneumonia, acute respiratory disease syndrome, multi organ failure, sepsis, septic shock, acute kidney injury, cardiac injury, and death. The very high level of pro-inflammatory cytokines, that is the cytokine storm or cytokine release syndrome, may be pivotal in the severe pathology of COVID-19. As mentioned before, tumor necrosis factors interleukin 1 and chemokines are the principal cytokines involved in recruiting blood neutrophils and monocytes to sites of infection. Tumor necrosis factors and interleukin 1 also have systemic effects, including fever by acting on the hypothalamus. These cytokines and interleukin 6 stimulate liver cells to produce various acute phase responses such as C-reactive protein and fibrinogen, contributing to the microbial killing and walling off infectious sites. At high concentrations, tumor necrosis factors promotes thrombus formation on the epithelium and reduces blood pressure by a combination of reduced myocardial contractility and vascular dilation and leakiness. Severe disseminated bacterial infections sometimes lead to a potentially lethal clinical syndrome called septic shock, characterized by low blood pressure, disseminated intravascular coagulation, and metabolic disturbances. The early clinical and pathologic manifestation of septic shock may be caused by high levels of tumor necrosis factors produced in response to the bacteria. The inflammation outcome depends partly on the extent of damage and partly the type of tissue damaged and the type of cells present in the inflamed tissue. Ideally, an inflammatory response will clear the source of damage or infections or toxins, leaving little tissue damage and minimum scarring. Scar tissues will form if the damage is extensive or if the tissue cannot regenerate itself. Scarring occurs after damage to the heart following myocardial infarction and, in some cases, pneumonia, where lung fibrosis can result. While neutrophils characterize acute inflammation, chronic inflammation is marked by the presence of macrophages and lymphocytes. There is always tissue damage with chronic inflammation and repair processes resulting in scarring. Chronic inflammation causes several conditions, including asthma, eczema, rheumatoid arthritis, and ulcerative colitis. Antibodies are differentiated into five classes: IgG, IgM, IgA, IgD, and IgE. IgG, these are the most commonly circulating antibody, about 75% of the total immunoglobulins. It is found in nearly equally equal proportions in the intravascular and uh, in, interstitial compartments, and has a long half-life of about 3 weeks. IgG is the smallest of the immunoglobulins and can more easily escape the bloodstream to enter the interstitial fluid surrounding tissues. IgM accounts for about 10% of the circulating immunoglobulins and is predominantly found in the intravascular pool. Its large structure prevents it from migrating through the capillary walls. IgM has a half-life of 10 days. It is the first immunoglobulin to be produced on exposure to antigens or after immunization. It is the major antibody found on B cell surfaces. IgM is the antibody class that works best to activate complement, which is important for cytotoxic functions in the immune system. Only one molecule of IgM is needed to activate complement, whereas two IgG molecules are needed to activate complements. IgA is produced by plasma cells located in the tissue under the skin and mucous membrane. IgA is primarily found in saliva, tears, tracheobronchial secretions, colostrum, uh, breast milk, and gastrointestinal and genitourinary secretions. Transport of IgA into secretions is facilitated by binding to a secretory component produced by epithelial cells. The half-life of IgA is about 6 days. IgD are bound to and they activate B cells. IgE are bound to mast cells in the skin and mucous membranes. They stimulate mast cells to release histamine in an allergic immune response, leading to inflammation. IgE has a half-life of 2 days. The sequence of events of acute inflammation shows three main processes, and these are: number one, the release of inflammatory mediators with the release of chemicals such as histamine, prostaglandin, bradykinins, leukotrienes from damaged cells, and immune cells, which trigger the vascular and cellular response. Number two, the vascular response of the blood vessels that promotes vasodilation and increased permeability. These become more porous and leak plasma and plasma proteins. Some of the proteins include complement, bradykinins, and clotting factors. The exudate carrying with it is bacteria, cell debris, and immune cells are drained into nearby lymph vessels. These activate the lymphocytes and initiate an adaptive immune response. Number three, in a cellular response, immune cells, particularly macrophages, mast cells, and neutrophils, play a vital role in inflammation. Chronic inflammation will arise with persistent infection where the microorganism has not been removed. An example is Mycobacterium tuberculosis, which can survive and multiply inside macrophages. Allergic diseases such as asthma and hay fever are continual exposure to the chemical causing the allergy or the allergen. Autoimmunity, in which the immune system targets the body's tissues. Prolonged exposure to potentially toxic agents, exposure to asbestos and silica can cause chronic inflammation to the lungs, as can cigarette smoking. Cholesterol builds up in the lining of large arteries, contributing to a chronic inflammatory reaction. This is the underlying disease process in atherosclerosis, which is the primary cause of ischemic heart disease. And this concludes my highlights for week 6. I will see you in week 7. Thank you for watching.